Hello, I'm Rachel Kolodny from University of Haifa. This is really fabulous crowd. Really excited to be talking here. Thank you for inviting me. Oh, whatever. יותר גבוה. Okay. Okay, so the question we're looking at here is what are the global properties of protein space, of the protein universe? Okay, and here's like an image of the protein universe that we drew. It's nice, huh? This is like a really fundamental question. And the essence of the problem in forming this global view of the protein universe is that what we have are glimpses, local connections between proteins. We compare pairs of proteins or maybe of several proteins. But what we have are these local glimpses, and what we're interested in is the global picture of what everything looks like. And this is something we're really interested in understanding, because we hope that if we can look at the global picture of what the protein universe is like, it would improve things like our ability to search for proteins, our ability to compare proteins, and maybe protein design. We could understand evolution better. Okay, like everything is based on our ability to understand what the global picture is like. So this is a very old question, and many people worked on it, many scholars worked on it. And we start from the data, from the PDB, which is the database of all proteins of known structure. Common wisdom says look at domains rather than protein chains, because these are the evolutionary meaningful units. This is actually, this is common wisdom, but one could question if this is really the right thing to do, and I'll get back to that at the end of my talk, because this is what we're actually looking at now. But say, for now, we'll look at protein domains, and then organize the set of all these protein domains into a meaningful, ordered view. So the classics are, of course, the classifications of protein space, and these, the most important ones, are scoped by the Merzen lab, Kath from Rengold's lab, and recently Nick Grishin and his lab worked on a new one called ECOD. And in these, you classify all domains. So for example, here's a picture from Kath. You start with a three-group classification, to proteins that are all helical, proteins that are all beta sheets, and ones that have both. And then amongst the ones that have both, you further classify them into groups, and then you move down this way. And in this view, implicit in it, is that somewhere when you go to the low level, which in SCOP is called fold level, and in CATH it's the CAT level, but somewhere down there, it's islands, groups of protein domains that are similar to each other and not so similar to other proteins in the world. Okay, so this is what proteins are like. Of course, cross-fold similarities, or similarities between proteins of different folds, have been known for a long while. And even these labs now, in their current view of the world, show these relationships. It has been shown that there are these. And this was called the continuous view of protein space, as opposed to the discrete view that is suggested by these classifications. Okay, so because these have this rigid, discrete view, people have suggested an alternative view of looking at protein space, which are maps of protein space. So in maps of protein space, the protein world is described as points in a two-dimensional or three-dimensional view. Every domain is a point, and it's colored according to some property. And the points are placed such that the distance between them in this 2D or 3D world is representative of some meaningful distance. For example, the structural distance between the proteins. So the first to do these were Rengo and co-workers, and then Holm and Sander. And then there was a series of beautiful papers from Sang-Ho Kim's lab. This is the picture I show here, where, as you see, they colored them according to the SCOP class, which is all alpha proteins, all beta proteins, alpha/beta proteins, and alpha plus beta proteins. And they showed that they cluster in separate regions of protein space. And even we have a paper where we mapped proteins into maps. Where we mapped proteins, we created these maps. And our cool factor in the paper was that we were able to do really large data sets. Ours has 40,000 proteins. And this allowed us to look at the functional diversity of the proteins, of how many functions are supported by proteins of a similar shape. And then we saw this core of high functional diversity in the middle. That's where the TIM barrels lie, but not only the TIM barrels, other stuff lies there too. So we were happy about this. So these maps are like really cool things. The only problem with them is that they do not support non-transitive relationships. So when I say non-transitive relationships, I mean that if there's protein A that is similar to protein B, so it lies close to it, and then protein B is similar to protein C because they lie next to each other, then A and C have to be similar to each other. And this is actually not true in proteins. It's true in Euclidean space where we map them, but it's not true in real proteins. This is something that was shown recently by in a Bastola et al. Paper, but it's just true. And it has to do with the fact that we look at subsections of the proteins to say that two things are similar. And this is not only when you look at domain rearrangements. It's very true at the subdomain level as well. So because there's this issue with protein maps, people looked instead at protein networks. And the story I'm going to tell you today is about a network view of protein space. And in these, every protein domain is a vertex, a point in the network, and two proteins that are similar to each other are connected by an edge. And the first work was by Golan Yona, and then Dokleyan and the Shaknoviches presented the protein domain universe graph, PDUG. Then there was a paper by Zhang, and Skolnick and co-workers presented a network where they connected two proteins if they shared a structural similarity of some sort, and they argued that protein space is essentially connected, that any protein you can go via very similar protein structures to any other protein in the universe. Okay, and the reason I know all these details is because not so long ago, Neil and myself wrote a review exactly about these, about representations of the protein universe. So we mentioned all these and many more that I didn't mention here because there are too many of them. Okay, so when we went to look at protein space, and the first step was to identify similarities in protein space. So we looked at all proteins against all proteins, and here I'm marking them just by 5 representative cartoons, and this is the shapes along the backbone, so I'm showing some repetitive shapes, and we compared all against all. It's only the upper triangle because it's the same thing. And in any case where we identified a similarity, I placed an asterisk. Okay. And then what we really did was not just 4 protein cartoons, but a database of almost 10,000 scope domains where we compared all against all and identified all these similarities. And then we drew a network where every protein was a vertex, a node, and every two that are connected to each other, for example, these two share this pink circle, we connected them by an edge. And this is our domain network. We did one more thing. When we represented every protein domain by a vertex, we colored it according to its scope class, which is another way of saying that we colored it according to its biophysical properties. That is, some are all alpha, alpha slash beta, alpha plus beta, and all beta, according to this color scheme. And we looked at the network that comes out of this. So here's a more concrete example of 4 domains that we looked at. And I listed between every 2 domains that share a significant part. For example, these 2 have 86 residues, which can be superimposed to less than 2 angstrom RMSD, and they have a 30% sequence similarity. This is very similar. It's really similar. One could argue that at this level of similarity, you could say that the 2 are connected, have an evolutionary relationship between them. And we use these thresholds, and this is the domain network that represents these 4 connections, these 4 domains. And they're all colored in green because they're the alpha slash beta proteins. Okay, so in this example, which I showed, we added an edge if there were more than 75 aligned residues that could be superimposed to less than 2.5 angstrom RMSD. These are very similar structures, and with a sequence similarity that is greater than 30%. Of course, you could use other thresholds. For example, these are the thresholds we used. We used, instead of 75 aligned residues, 55, and instead of 2.5 angstrom RMSD, 2 angstrom RMSD, and so forth. And I'll talk in a minute about how we selected the thresholds that you actually use. But there is, I want to highlight that if you have the alignment, if you identify the 2 sets of substructures in the 2 proteins, checking what is the RMSD or how long the alignment is or what is the sequence similarity, this is a trivial process. The challenge is to find these alignments. But we didn't do it. We actually used the structural alignment method called SSM, which was shown to be particularly sensitive, and they found the alignments for it. Do you want me to turn the
I'm fine now.
Okay.
The rest, I don't know, but I'm fine.
Okay. Cool. Okay, so we use this. So because you could argue that different thresholds are the good ones here, you could do one of two things. And we actually did both of them. So the first is to say, let's look for the perfect threshold, or a good threshold, or set of thresholds. And I'll go down that path in a minute. But before I do that, I want to mention the following thing. If you use very lax thresholds, you will get a very connected network. This is basically what Kolodny did. He used only structural similarity. That means that any sequence similarity was fine. But then everything is connected to everything. We actually showed that if you use 55 residues, 3 angstrom RMSD, more than 25% sequence similarity, it's one connected component. And this here I show a slightly stricter set of thresholds, but it's really everything is connected to everything. This is expected. If, on the other hand, you use very strict thresholds, here, for example, 75 residues, more than 50% sequence similarity, then it all disintegrates, right? You would expect that to be the case. So the two endpoints are trivial ones. But you could think of these as if you're going further or closer in evolution. If the thresholds are super strict, it means you're just looking at yesterday. And if the thresholds are very lax, you're looking further away in evolutionary time. And this ability of changing the thresholds gives you a hint of what it may be, how it disintegrates in evolution, right? So maybe you could vary the thresholds and look at all the pictures that you get and get a picture from that, from the varying the different thresholds. Okay, but before that, we tried taking the, I think, more standard route of finding good thresholds. And the way we wanted to find good thresholds was by calibrating them to scope folds to the existing classification. So what I mean by that is the following. When we disintegrate, then stuff that falls in the same scope fold no longer is in the same scope fold because the network disintegrated. So we would like to vary the threshold and exactly to the point where the folds, where the folds are separate. Each fold is exactly well defined in the network, is a connected component. Okay? So what we plot here is for different thresholds. For each one we checked among every fold how many cases we have that pairs that are in the same fold are indeed within the same connected component, as opposed to how many pairs that are in different folds but fall in this, the number of failures on success in placing in the same connected component when you should, versus the number of failures in not placing them when you shouldn't. Okay? So first I want to point out that there are 2 points in this graph that are completely trivial to achieve, which are this one and this one. So what I mean is if you want a complete success, 100% success, in placing the same connected components within the same scope fold, what you can do is use very lax thresholds. Everything will be a single connected component. And in every single case where two things are in the same fold, they will be connected because the whole thing is connected. Alternatively, being completely successful in not placing them in the same fold when you don't want them to be in the same fold, what you could do is use very strict thresholds, disintegrate the network completely, and then it never happens that two things that shouldn't be together are together because everything is apart. Okay, so these 2 points are really trivial. This is not, there's nothing to talk about. But this point of achieving the two of them at the same time, this is a challenge. To get there, you need a good threshold. So what we're looking is for thresholds that are closest to this corner point. And you see, this is the one I showed you. But this is the good point, which is 2.5 angstrom, 75 residues, and 30% sequence similarity. I'll mention in a word that the thresholds that you use actually have to do with how good your search procedure for alignments is. When we used a different search procedure of the HH search of a sequence aligner instead of a structural alignment, then the thresholds were different because the search procedure was not as sensitive, was not as powerful, so to speak. So we needed less strict thresholds. Okay. So we found that one, and then we drew the network, and this is the network that we drew. And it's amazing. I've given this talk several times by now, and I've looked at this picture a lot of times, and still every time I look at it, it shows up, and I'm like, whoa. It's still really a nice pic. It still makes me happy because it's an amazing picture of what protein space is like. So the striking thing about it is that there's a region where the air conditioner is excited. There's a region where protein space is connected, like it's one big connected component. And there's a region where every fold is a different connected component. And there's two types of regions in protein fold space. It's not the same. Where the alpha/beta proteins lie in green, the big green bulb, everything tends to be connected to each other. While with the place where the all beta, the alpha plus beta, and to some extent the all alpha proteins lie, then they're separated into separate folds. And this is like the very long debate of what is protein space like. So in some sense, if you're looking at the proteins, like the all beta proteins, the classification such as SCOP it's an excellent approximation of what protein space is like. When it groups them into these folds, it describes it like really well. But in the alpha/beta proteins, where the alpha and the beta iterate between each other, this is actually not such a good approximation. Let me show you. Here I colored the same map. I colored some SCOP folds, like not all of them to make the picture not too heavy, but I colored some. So for example, you see that the globins, this is like A1, this is one fold. And here's another fold. This is the D131. It's the alpha plus beta protein. Or the orange, which is the D38, right? There are many folds. This is exactly where all these folds lie in one connected component. But here you see the timbales, which are closely connected to the P-loops. Like they're all connected one to the other. And of course, they lie next to each other. The folds are, of course, the domains in one fold are all similar to one another. But they're also significantly similar to to domains in other folds, not just to domains in their own fold, which is, okay, it's evolutionary related. These are strict thresholds. The structures are similar. The sequences are similar. It's over a significant number of residues. It's more than 75 residues. Okay, it's like this. Okay. So one could wonder if the issue is that it's not really that the thresholds, it's not really that there's regions in protein space that are far more connected, the alpha/beta, but rather it's just different thresholds. That somehow because of the structure of the alpha/beta or because of the sequences that accommodate the alpha/beta protein, it's just different thresholds. And if I would have moved them around more, I would have disintegrated that region into folds as you would expect it to be. So we tried doing the same process for each one of the SCOP classes separately, just to see if it's different thresholds, if this is the issue. And what we saw was, okay, so this is what we saw. If you look at the all beta proteins, for example, you see that you can really find thresholds that are like our threshold, the one we mentioned, that really finds points closer to the sweet corner. This is really happening. And similarly, the alpha plus beta to some extent. But if you look at the alpha/beta, there is no combination that can separate the folds into separate folds, into separate connected components. Or another way of saying it, when you disintegrate the network, it does so instantly. Or, right, it doesn't happen that you separate it and it all falls apart in one round. Or another way of saying it is that the similarities within a fold is on the same scale as the similarities between folds. Okay, it's all saying the same things. And in the alpha/beta, this is what goes on. Like there's no way of avoiding this observation. And in the alpha plus beta, less so. And right, so this is the most fold-separated one, the most discrete region of fold space. This is the most continuous region of fold space, and this is more to the continuous, and this is more to the discrete. The all alpha is more to the continuous, and alpha plus beta is more to the discrete. So, okay, so this was the. So here's an example from cross-fold similarities that we observed with these thresholds. Here I took a path within this domain graph over 5 different SCOP folds. Okay, so I'm starting with the timbales. I'm crossing over to the C23, C2, C4, and C3. And as you can see, in each case, I highlighted the similarities. These are really, I'm trying to push the point that these are significant similarities over many residues. The structures are really similar. The sequence similarity is high. Okay, this is, I'm just trying to reiterate this same point. But another reason I'm mentioning this is that this is actually an interesting question. If you could walk around protein space and look at how things are connected to each other. We spent a lot of time doing that, walking around protein space and looking at if this is similar to this. Oh wait, and this is similar to this. And like moving between them and looking at these relationships, which we think may all have some evolutionary significance. So when we did this, we spent a lot of time doing this, we used this tool called Cytoscape, which is an open source, really cool network visualization tool that we used a lot. And because we kept looking at the structure, Sergei Nepomniachtchi, who was a student of ours, did this thing that he connected the molecular viewing capabilities of the molecular viewers. We were using PyMol at the time, but because he did such a general job, he actually connected it to all molecular viewers, and he connected it to Cytoscape, to this network visualization tool. So I wanted to just mention these things that he did, because everything is open source and you can download it. So because we have this cool network visualization tool coupled with the molecular viewer, all our data is online, all our domain networks, all the alignments, everything is online. You can just download it and walk along the network and see it. So the way Sergei did it is that if you click on a specific node, which is a domain, it opens and you install our plugin, which is called Cytostruct. You get it? Cytostruct. So if you install it, and it's a really cool thing to install, I'll mention it in another slide, you can right-click and get a menu which opens Cytostruct, and then you can open it in a molecular visualizer. You can do it either from a node or from an edge. In case of an edge, it connects two nodes, meaning that it's two different protein domains, and they're superimposed one on top of the other. You see? They're superimposed, and then you can look at one of them or look at the other within the molecular viewer, within PyMol. And actually, the superpositioning of the two domains is done within PyMol. The reason I'm mentioning it is because the molecular viewers, PyMol, VMD, Chimera, all of them have like significant computational capabilities. They can do a lot of stuff. And the way this is implemented, Cytostruct, is that you can use anything that the molecular viewers can do for you from the network. All it does is generates a script. So I'll show you this in a minute. So at some point, because Sergei was so keen on doing it in a way that he wouldn't have to configure anything, and it was just this general purpose tool, and it was configured from the outside, it turned out to be a very useful tool because it can connect any network to any molecular viewer. So what we noticed is that we can visualize complexes, molecular complexes with it as a network. And we did it. Here's an example. Oh, wait, let me say it. Here's an example of how we visualize this network. So here's the structure of 3J7Y, which is a large ribosomal subunit of the human mitochondria. It was published in Science last year. I'm sorry. Okay, so Sergei visualized it as a network. It has 48 proteins. So these are 48 nodes, and they're connected to each other in every instance where the two protein chains have an interface which is greater than 100 angstrom square. This is calculated by another piece of software, which is not ours, PISA, by Henrik and Chrisino. And when using Cytoscape, you can, in every instance that you click here, you can see the two molecules within any molecular viewer. We implemented it with PyMol, UCSF Chimera, VMD, JMol. They kept adding them, and we kept, there's no issue doing it. Okay, and it's all online. You actually even implemented an online version. You can give it any complex, and he will generate a viewer with this molecular viewing capabilities. So I just want to mention that the way he does it is the network is in Cytoscape. And then within Cytostruct, you configure two things. The first thing is you configure the template script, which has pieces missing, and these depend on the node or on the edge. And then you configure a data matrix, which has the edge and node specific data. And then Cytostruct infuses the data from the data matrix into the script and runs the script. So anything, right, it's a very straightforward idea. It's like there's really nothing to it, but it's super powerful. Like we're using it all the time, and anyone who is interested, just talk to me. Like it's all online. The code is in Bitbucket. It's examples, demos. It's all there, and you're welcome to use it. It's really super cool. So if anyone has and because it's so general, it doesn't have to be actually a molecular viewer. It can run anything. So if you have a program that does something else, but it can be scripted, it could be used. So anyone who has something like that and would like to configure Cytostruct to use with it, then come talk to me. I'll be happy to walk you through it. Okay, so here's another thing that we used with, Cytostruct because we already had it. So I'm slightly switching, but actually it's the same, in some way it's the same thing. So this is a work that Neil Bental's student Aya, who's somewhere in the audience here, did, and it's called Contemplate. So what Contemplate does is it suggests possible alternative conformations for a protein structure query. So this is the idea. Proteins often appear in multiple conformations, and even in the PDB you find often proteins in multiple conformations. So if you have a protein query and you know its structure, you can ask Aya's server, Contemplate, to find for you additional conformations that appear so you can find first proteins that are similar to your protein, and then there are alternative conformations, and these will be suggested as alternative conformations to your protein. And in cases where the two proteins not only share one conformation, which is why you found them, but share the other conformations as well, then this would be a useful piece of information. Okay, so this is what Contemplate does for you. And this is, of course, online, and it's all supported. So here's an example of what it would do. For example, if you have a query protein, this is a ribose binding protein, and this is the, starting from the unbound state, this is your query protein, and then you can find another protein that is similar to it, that's the green. This one has an alternative conformation. No, the similar protein is this one. This one has an alternative conformation, which is the green conformation. And if you look at it, you see that the green and the true alternative conformation, which is the non-bound version of your protein, which of course Aya took out of the database to find out about this, you find it, and you actually find a very good approximation. She models it with Modeler. This is a 1.65 angstrom RMSD. But the two conformations, they vary by more than 4 angstrom. These are significantly different conformations, and Aya can find them for you. And of course, it can walk the other way backwards. Like you can start from this one and get to that one. So I think about it as, I think of Aya's server as a way to zoom in on regions of protein space, right? She looks for you for proteins that are similar to your query protein, and then for these she looks for their alternative conformations. And this is a little piece of protein space that is relevant to the person looking at this problem. So she models it as a network in her results. She gives you back this view, this network view of protein space that is relevant to you, this little region of protein space. And in this case, for example, this is the target structure and this is the closed structure. And oftentimes it happens that many of the different proteins in one conformation, in this case the closed, there are many of them in the PDB, while the other one has just a few. But okay, this is the case. And in this case, they modeled it so that the edges are, the length of the edges depends on the structural distance between the two conformations. And of course, this is implemented with Cytostruct as well. And then if you right-click on it, you will get the structure, the superimposed structure. Okay. So, okay. So I mentioned that if we have all against all protein comparisons in this network, you can compose the similarity network, which is what I talked about till now. But actually, another thing that you could do is you could talk about the co-occurrence network. So let me say a few words about this. So in the co-occurrence network, what you could do is you could identify here pieces that are recurring in protein space. They're often called motifs. For example, you see that here the pink diamond reoccurs in 2 proteins. So this is a motif. And you could draw a network which has these motifs. Actually, from this data, you wouldn't be able to detect the purple heart because it appears actually only once. You do not see that it reoccurs. Okay, but the ones that are reoccurring, you could identify them. And then in every instance where 2 motifs are co-occurring in a protein, for example, the yellow triangle and the pink diamond appear in a protein. So in these cases, you can connect them by an edge. So this is a network that is very close to the dual network of the similarity network. It's a variant of the dual network. But okay, it's a co-occurrence, and people have done similar work on co-occurrence of domain entities in protein space within the same chain. But you could look at the subdomain level and do this similarity, this co-occurrence network. And actually, we did a bit of that. So, for instance, in this case, you have three motifs: the cyan one, the orange, yellow one, and the green one. And you can see, right, there's three motifs, and you can connect the yellow and green one by an edge because they co-occur in D1, 0, 2, 0, 8. Okay, so this is the motif network. Okay, so the reason I want to talk about, the reason I mentioned it is that one would like to ask, how do we find these motifs? How do we detect these motifs from the similarity network? So it's obvious from this picture that there are many cross-fold similarities, many relationships between proteins, that there are many instances of these motifs that are shared between two domains. These are very common. The question is, what is the size of these motifs? This is like a, so now I'm talking about, like, at the end there's an algorithm that we already did, but before the algorithm I'll talk a bit about the motivation. It's the first time I'm talking about it, so I'll resort to talking about it in metaphors as opposed to data. So I hope this one flies. But it's something we're excited about, so let's hope for the best. So, so the question is, what is the size of these protein motifs, right? And when people talk about domains as these recurring elements, what they're suggesting is that these are the domains, or the recurring elements that are meaningful. But maybe they're smaller than the size of the domains that we have now in the current databases, right? It's like a valid question of what is. Of course, there is some trivial size of the amino acids. Like if we keep going down, there's the recurrent. But the question is there something in the middle, in the mid-range, that is recurring? And obviously, there's a lot going on with similarities. The question. Okay, so for this thing, let me resort to this metaphor. And imagine that let's just focus on two folds. One is a cat-like fold, and one is a lamp-like fold. And within, like, if these are the two folds that we have, we see multiple instances of cats and multiple instances of lamps. Okay, like the lamp-like fold has 3, and I did it with a different shade, and the cat-like fold has 5 different folds in space. Okay, but it could be that actually they're composed of smaller entities. And here I use the Lego metaphor, right? That the cat and the lamp are actually built from Lego. It's amazing. You can find these online, right? You can just go and write cat in Lego and you find these pictures. So maybe the cat and the lamp are built from Lego building blocks. And in this case, it's actually smaller common motifs, right? Like it's actually, and it's a question, is this the picture or this the picture? This is the, so what I'm pointing out here is an observation that we had, is that if you would look for redundancy at smaller levels, at shorter size pieces, for example, if this is the world and you cut it up into smaller pieces of recurring elements, for example, you would cut the cat into 3 pieces and the lamp into 4 pieces, and then you would measure again the redundancy that you have in protein fold space, you would get the same redundancy level. You would have more pieces, but it would be the same redundancy level, right? Like you have 5 copies of this right-hand side of the cat. But if the cat and the lamp are made of Lego parts, and you check the redundancy, what you would get is a pile of Lego building blocks, and these would be far more redundant. So this is what we want to do. We want to check the redundancy level at different levels of sizes for the proteins and check what is the redundancy, and this would give us a hint into what the real size of the motifs in the protein world. And of course, it could be that it varies in different regions of protein space. This is actually what the continuous region versus the, presumably in the regions of where it's a fold, the redundancy level is just the domain level, right? But in the alpha/beta continuous region, it's different. So to do that, what we need is a way to succeed in detecting motifs from alignments, right? This is actually, I think, this was the issue and why this was not done thus far. So you need an algorithm to detect motifs from alignments. And this is what we have. So just like they say in the $6 Million Man, we have the technology. So I'll just spend just a little bit of time to describe how we do that. So what we start from is from looking at all the alignments in space. But actually what we do is we focus for every single protein domain on all its alignments to other proteins. So we're looking at a protein and all the things around it, all the alignments that we found for it. And again, we're enforcing alignments that we trust, ones that have a significant number of residues and that the sequences are similar and that the structures are similar. But here, we don't have to go for really long alignments, right? If it's just longer than 35 residues, it's fine, because we're actually interested in the shorter motifs. We don't want it to be just 75 residues or something like that. So we did that. And of course, there's the question of how do you find these alignments, not just how do you verify them. And actually for this, we tend to use the sequence alignment rather than the structure alignment, which is less sensitive but more conservative in nature. You find things that are really, you trust them that they're evolutionary related. This is why this is a very successful but profile sequence alignment. This is why we went back to HH search. Okay, and then you have a protein sequence. This is protein 1, and all the alignments that it has to the other proteins. Of course, it will have gaps. It's just for the sake of the cartoon. I drew it without the gaps in the first one. So let's just cartoonify it. So we have a protein in gray, and these are all the alignments to the other ones. Okay, and what we want to do is to chop protein 1 into motifs, which are recurring. And we want to do it in an optimal fashion. So what I mean is, for example, if we look at this motif and this motif, I want to highlight that these are quite good ones because each one appears in 4 different other proteins. It appears, right? Well, if we looked at this motif, it's just in 3. And if we looked at this motif, right, it's less successful in modeling something that is recurring in protein space. So what we would like to do is identify these motifs computationally, like not look at a case like this. And the way to go about it, it's actually very straightforward if you think about it. So if you have a protein aligned to another protein and you're considering a particular part and saying, is it recurring in the other protein, you can just measure how, you don't say, yes, it reoccurred, these sequences are aligned. You would say how well it reoccurred, and you measure it with a scoring matrix such as BLOSSOM. You have a score for every, for this little piece, you can measure how well each one is similar to the other, and you can sum the score. And if you have multiple sequences, you can sum the score over all of these. So what you would want to do is you would want to find a partition that maximizes the score, but is longer than some threshold, say some motifs of size 35 or motifs of size 40, or you can do it for different scales. And of course you could do it exhaustively, check all the scores, but that would be non-efficient and even worse, non-elegant. So instead we have developed a dynamic programming algorithm that would find the optimal one, but would do so efficiently. So we can do it for all the proteins in protein space. And this is what we actually do. So for example, okay, it's optimal under this modeling scheme, but just to convince someone who's not convinced in the optimality of dynamic programming, I'll just show you an example in this protein and find this motif and some others. I just colored 2 of them. Or, for example, oops. Okay, let me just say this first. So once you have a motif, you can go and search for that particular motif. It's a structure and a sequence in all of protein space and find all its occurrences in protein space, right? This is what you would want to go about doing. So you could either compare it directly to the PDB, or you could use our maps and just find where this motif occurs and look for it, like walk over protein space and search for that particular motif. So it would be slightly more efficient. And also it would be more conservative because the alignments connect stuff that the flanking residues of the motifs are aligned as well, not just the motif itself. Like this is a more conservative selection. And when we did this conservative selection, we found, so I'm just showing you to show an example that we can find motifs that are, it's obvious to anyone who has been looking at proteins that these are reasonable motifs. So the most popular motif we found was the beta alpha beta, the one in the alpha slash beta, and then we found this like a little piece of beta helix and from a beta blade from a propeller. Like it's really exactly what you would expect to find. But obviously the algorithm can do that. And of course, if we set a threshold that is very high, that is, if we're looking for recurring elements of more than, say, 100 residues, it will find back the motifs, the domains themselves, as you would expect. This is an automatic way of finding domain limits. Okay, so that's it. So this is what we plan to do. We didn't do it yet, but we have the algorithm, so now we can do it. So I hope I convinced you. I finished before ahead of time. I get extra points. Okay, so I hope I convinced you that the domain and motif networks are a useful way to study the protein universe. Like we found them illuminating, so that's why we told you about them. That protein space is both continuous and discrete, it's an important observation. Because the continuous regions mostly host the alpha/beta domains, which were noticed to be different from others, but this is another observation about how they're different. And that cytostruct can help in this case and in others. We think it's a really useful way of visualizing stuff. And we hope that people will use, and we will use our approach to theorize about protein evolution and the physical chemical properties of proteins. And maybe it will help to organize and search protein space better, and maybe even help protein design. Who knows? Never know. And wait for it. I want to thank you for your attention. It's the end of the day. I know it was a difficult one to listen to. Neil and Sergey, who's not here, did everything, all these with me. And also Aya, who is in the back somewhere, did the contemplate. And thank you for your time.
When you're doing this work, do you take the ligand, the binding, or things like that into consideration? Because it may change the structure of the protein, and actually there are some crystal contact can also have an impact on the protein folding, but there's no biological meaning. So when you're gathering this data, how do you manage that?
So it depends on when I look at, so for example, in Aya's work, in Contemplate, this is exactly the point of finding these, if you have the ligand-bound structure, if it could find for you the non-ligand bind, like the apo form. Right, so this is exactly what Aya is looking at. And actually, incidentally, we have a paper from a few years back where we looked at pairs of proteins that have almost identical sequence identity or similarity, like really similar proteins, the same chain basically, that have significantly different structures. So of course this happens in the PDB, but when looking at the, like there's like 10,000 structures here, like we don't want to overdo the redundancy. So of course we're missing some information, but I don't think we're missing the big picture. I think there are places where we're missing some information, but for the big picture, of course we're taking a 40% non-redundant set. So because if we took a more redundant set, we would be missing the picture from a different direction. Like we would have these hyperversions of some of the structures that it's not that there are more of them, it's just that more of them were solved. So like we're trying to compromise between these, but we took a 40% non-redundant, and of course we know we take the best crystal structure, like typically non-redundant sets take the best crystal structure, and we only looked at crystal structures, like nothing that is non-structural. This is the standard way of doing it, like we didn't invent anything in this.
Do you see, or have you looked at evolutionary connectivity between like functional units, for example, respiratory or mitochondrial, something which also things that are supposed to work together, or proteins with the flagellum? That's one question. Another question is, you see like hyaline, for example, member proteins form their own.
We don't have any of those. The structures are not good enough. No, but the reason we're talking about this is actually, like, we were really thinking of the question of how do you look at protein space. Like, we started from a very naive question of what's it like. Like, this was the level of the question that we were looking at. And it turns out that even at this very naive question, because like all of a sudden you're using your cognitive abilities to look at it, you get these insights into questions we've been asking for a while, but couldn't quite pinpoint what the thing is. But what we think about these maps with the molecular viewer incorporated, like with the whole thing, what we think is really useful about it is that it can be used, like you could overlay it with any type of information. And of course, functional information is what would be super cool to overlay. So for its first step, what we overlaid it was just the most trivial information with the scope class. That's like the biophysical. And then like a little bit of scopefold just to see that it makes sense. But of course, what we care about is the function, and Neil has a student, Shai, maybe he's here, he is, that started doing this. And also we're hoping of incorporating this whole view into ECOD. And we're hoping that there the functional information would somehow, but of course this is why we think this is a useful thing, not just because of this. Like, of course we were part of this argument, is it continuous or discrete? So we feel that we kind of won. But but this is not the reason this is interesting. It's really interesting because it gives us a way to look at, to reason about this business of protein spacing. Yes, Alva et al., of course. So it's a beautiful paper. You're referring to Alva et al. From Lupash, Soding, and Alva. And they did a visualization that is similar to this. They didn't look at the structural similarity. They only have HH search similarities between the things. And somehow they missed this bit of the continuous versus discrete. Like they were almost there. Yeah, they just passed next to it. But it's, of course, we read their paper and we appreciate it.
I have a second question. How do you handle with, there are some examples of proteins that have dislocation, meaning that you can find them at sequence level, but in structure level, they present this dislocation. Have you considered them?
Yes, no, but we were conservative about it. So what we did is when there were two pieces, two subsections that were aligned to each other, we verified that it's big, because sometimes it's small. So we threw all the small ones away. And then we enforced that the sequences will be similar. And then we enforced that the structure, and only if all three conditions apply, we added an edge. So this is a conservative picture. There are many connections that are not seen. And the reason we took this conservative picture is because we wanted to be like the sequence aligners. When they say two things are similar, sometimes the structure, sometimes it's just a motion, but sometimes it diverges more, and then I suspect the sequence aligner were not, like they went for the more sensitive, but they lost accuracy. So this is a way of being conservative to make sure that these are evolutionary related things. And of course you miss some, but this was actually on purpose. We were trying to be cautious.
I have a question about finding the motif within this network. I think I understand that if you have a given motif, it's at least conceptually it's straightforward to find it whether it exists in other proteins. But how do you know which motif you are going to find? How do you start with this motif?
No, so I meant to explain it, but let me try again. So the challenge is, what we say is we have this protein. Let me try and use an analog. If you look at people, and you look at a family of people, like a particular family, and you notice that all of them, if you compare them to each other, and you tend to see that the nose tends to be the same between different people, then you can conclude that the nose is a recurring element. So or if you compare pictures, and you see in different pictures there's a dog, and in different pictures there's a car next to it, you could conclude that there's a car element and a dog element just because they repeat in different pictures. So here we're trying to identify pieces that happened over and over again. And what we're trying to find is we're trying to take the proteins and chop them up into pieces such that the pieces, it was that they occurred most of the greatest number of times in other proteins. And we saw that they occurred the greatest number of times because we started from alignment data. It's a real challenge. This is not like we've been futzing with this forever. Like it took us a while to find this. It's non-trivial to identify these pieces because the alignments are not this clean looking. Like to find an optimal.
How big are they?
No, so you can say, I want only alignments that are greater than, it's a parameter of the algorithm, and this allows you to find redundancy at different lengths. And then you can compare the different lengths and reason, like in this region of protein space,
The definition of the motif depends on...
Yes, because it's dynamic. Yes, so let me say it another way. You can, when you say, I want to look at all the motifs that are 35 residues or more, you could exhaustively check all endpoints that are starting from 35, right? It would tell you for some of the endpoints. Of course, you could compare different ones, like you could look which one got the best scores. But you could even say, I don't need to check the ones that are shorter than that. It would make your algorithm more efficient. If you're penalizing gaps, it would tend to go for the shorter ones. You could, of course, penalize it less, but it would tend to go for the shorter ones if you're penalizing gaps.
Does it get you to the kind of prototypes of, let's say, CAF at the level of the architecture and so on?
Yeah, the domain, yeah. No, it does. So this is it's a good question. So we asked that because this is like an automatic domain definition system, right? So first of all, they don't use only recurrence. They use globular and shape. But we do get stuff. The cases we checked are so on. The problem is that Cath and Sculpt, they don't agree with each other, and they don't agree with ECOD. And this is like this is one pit. Not everyone has to go down. Thank you.